Can you hear me? All right. Let's get started. We are here for the PhD defense of Elizabeth Knorr. I'm Ben Diel, I'm Liz's PhD advisor, and it's my pleasure to welcome you all here today, including members of the public, students, faculty, staff, collaborators, and Liz's friends and family. So Liz was born and raised in Sodus, New York, which is a small town near the South Shore of Lake Ontario. She received a Bachelor of Science degree from Hobart and William Smith colleges, where she graduated cum laude with a double major in biology and environmental studies. Liz started her PhD at OSU in 2016, and when she arrived here, our lab was an empty room. Knowing we had shared interests in disease ecology, I said that Liz should work on whatever she wanted. So while her classmates were out getting to know their field sites and lab protocols, Liz was staring into the maelstrom of our discipline, tasked with designing a successful project from zero. Fast forward a few years, and Liz had established a brand new empirical system for studying host microbe interactions in social populations using methods she pioneered for our lab. Liz has grit and an extraordinary level of follow-through. She is so steady in her work, and I think that steadiness is one of the most powerful traits that you can have as a scientist. Liz is also very steady and kind in her interactions with colleagues and mentees. She's mentored several undergraduates in her research, as well as other graduate students in the lab. And lately, I found myself responding more and more to questions from folks by saying, ask Liz, knowing that that was the best possible path to a good answer. So Liz, you will be missed. I remember a meeting that Liz and I had this summer, and the purpose of that meeting was generally to look at some challenges she was having in the post-PhD job hunt. Over the course of our conversation, Liz quietly led me to the root of the issue, which was she had like 8 postdoc offers, and I didn't know which one to choose. And I'm so proud to share that Liz will soon begin a prestigious APHL CDC infectious disease fellowship at the Wadsworth Center for Public Health in New York State. And there she'll work on quorum sensing in bacteria and specifically on Pseudomonas infection in patients with cystic fibrosis. Liz, I'm very proud of all your successes, and I'm honored that you were my first PhD student. And I thank you for taking the chance on a green assistant professor. And I'm looking forward to our future papers together. And with that, I'll pass the mic to Liz, who is going to talk to us about structure and dynamics of the canine nasal microbiome.
Okay, thanks, Ben. And thank you to everyone who's here today. I'm excited to share what I've been doing over the last 6 years. So today I'll be presenting my dissertation work on the structure and dynamics of the canine nasal microbiome. So first, what is the microbiome? One definition is that the microbiome is the diverse collection of microorganisms living in and on an environment. And specifically for the purposes of my research and this talk, we're interested in the non-pathogenic bacteria living in the host canine nasal canal. And so we know that there are these bacteria living in and on all of us, but why should we care about it? Well, the microbiome and hosts are strongly connected and have a commensal or mutualistic relationship. And the microbiome provides a number of host services. For example, the gut microbiome plays a crucial role in metabolism and nutrient acquisition by converting inaccessible nutrient sources into metabolites that our body can absorb. The microbiome also influences hormone release in the gut, affecting downstream metabolic processes, and all of this has consequences on host health. The microbiome is also important for immune regulation. The immune system needs to maintain homeostasis with the healthy bacteria, but also be able to recognize pathogens and prevent infection. So one mechanism is through the production of molecules like short chain fatty acids that can modulate host signaling, leading to changes in host immune cell activity. Commensal bacteria can also regulate inflammation through signaling and sensing immune cells like T cells and dendritic cells. The microbiome is also important in disease prevention. So the mucosal epithelium is a vital barrier against pathogenic viruses and bacteria. For example, the healthy bacteria in the microbiome can influence mucus production, providing a physical barrier from invading pathogens. Bacteria can also regulate mucosal permeability and produce antimicrobial and antiviral compounds. And proper priming and signaling with the immune system can allow immune cells to target invading pathogens. The microbiome is closely linked to host health, and changes in the microbiome during disease has been found across host organisms and body systems. So dysbiosis is the broad term used to describe any change from a healthy microbiome and can be defined as the imbalance of microbiota associated with an unhealthy outcome. And dysbiosis of the microbiome has been found related to obesity, gastrointestinal disorders, periodontal disease, pneumonia, asthma, and respiratory tract infections, among many others. So there's a clear connection between the normal healthy microbiome and a microbiome during illness. There are also several factors influencing the composition of the microbiome and differences found among hosts. So for example, body sites seem to have specific assemblages of bacteria, like those found here among the body sites on dogs. Immune interactions can structure the microbiome. Diet has been strongly linked to gut microbiome, and changes in diet can quickly lead to changes in microbial communities. Genetics, lifestyle, antibiotic use, and close proximity to other individuals can modulate your own microbiome. For example, unrelated people living together have more similar microbiomes than they would have with their siblings that they don't live with. Thinking about the different factors that can affect microbiome composition, my research focuses on pathogens, host traits, and microbial interactions. So a roadmap of my talk today, I'll provide some more background on my research and the canine nasal microbiome specifically. Then I'll describe my 3 research projects. First, the relationship between the pathogen community and canine health and disease. Then moving to the role of the host in nasal microbiome structure. And finally, chapter 3, exploring potential microbial interactions within the microbiome. And as a guide throughout the talk, you may see this bar on the bottom or any of these symbols in the corner to kind of remind you where we're at. Similarly to macroecological systems, the microbiome is a diverse community of interacting species with host health as an emergent property. So for my research, I propose the use of community ecology frameworks to generate hypotheses about how these communities are assembled and what forces can change them. Because really, the questions about how the microbiome impacts health and disease are similar to ecological questions underlying species diversity, invasion, and community stability. Community assembly theory can help us understand processes shaping the microbiome. For example, under this framework, there is some regional species pool that's a collection of all the microbes. And through dispersal, these microbes can establish within hosts forming local communities. But environmental filters can prevent proper establishment in all local areas. So for example, as an infant, the microbiota from your environment and from other individuals you may come in contact with could be your regional source pool of all the microbiota that could colonize your body. But environmental filters like a hot or dry temperature might kill some bacteria in the environment before you contact them, so colonization can't happen. Or your immune system is still developing, and so they aren't able to establish. And then in your local communities, species interactions through forces like competition between microbes for resources may shape what bacteria are living there. In these ways, the microbiome composition can be determined through community assembly principles. Metacommunity theory can also provide a useful framework for understanding microbiome dynamics and in particular, compositional variability within and between hosts. So each individual can be viewed as this kind of island-like patch of habitat occupied by these microbial assemblages formed through the fundamental processes of community ecology like dispersal, environmental selection, and ecological drift. The predictions of community diversity, abundance, and composition depend on the frequency and extent of dispersal and the degree to which these patches vary in their environmental conditions, potentially depending on host traits. Particularly relevant to my research is the prediction that microbes can disperse between hosts, and this dispersal may be aided or be more successful when hosts have closer contact. But host habitats are not equal, and differences in host traits can also affect the microbiome composition and which microbes are able to establish. For example, immune responses could impact which bacteria exist in the nasal microbiome. One way we can measure microbiome composition differences is using diversity metrics. So alpha diversity is the species diversity at a local site, while beta diversity is the ratio between regional and local species diversity, essentially comparing the similarity in diversity between two sites. So in my research, I use the Shannon Diversity Index to quantify the local diversity within a single dog. This measure takes into account the number of species living in a habitat, the richness, and their relative abundance or evenness. So how evenly spread amongst the different species the total population is. So for example, here community A would have a Shannon diversity of 0.5, while community B has a higher Shannon diversity of 1.39. And this makes sense because dog A has only 2 species with a large number of individuals in one species. While dog B has 4 different species and high evenness, meaning that they're found all at the same abundances. And dog C's Shannon diversity is between the others because it's more even than dog A, but not as diverse as dog B. For beta diversity, we compare the similarity between 2 samples or communities, and we use the Braid-Curtis dissimilarity, which quantifies the compositional difference between 2 sites by considering the shared species abundances relative to the independent site diversity. And the Braid-Curtis dissimilarity is always between 0 and 1, 1 being if communities are completely different, and 0 if they're exactly the same. So in our examples here, the Braid-Curtis dissimilarity between dog A and dog B would be 0.78. They only share that one triangle species. But when we compare dog A to dog C, they have a lower dissimilarity, so they're more similar because they each only have 2 species and multiple of these blue circles. So dog A's microbiome would be more similar to dog C's than it would be to dog B's. To understand the nasal microbiome community structure, I use a unique study system of dogs in the Oregon Humane Society Animal Shelter. And one reason this system and the Oregon Humane Society is so unique is because almost all of the dogs in my study were transferred from California and a few other animal shelters in Oregon up to the Oregon Humane Society through their Second Chance program. So basically, a truck goes and picks up these dogs from these shelters down here, and then they travel up to Oregon into Portland and arrive at the Oregon Humane Society, sometimes after over 8 hours of travel. So these trucks arrive a couple times a week, sometimes with over 20 dogs in them, and these dogs are then housed in kennels. And based on veterinarian staff observation, respiratory disease and other health problems are identified. And so when our dogs are housed at the Oregon Humane Society, there's a specific layout that's relevant for my research. So the layout of the shelter is essentially 4 large rooms called pods. And then within the pods are kennels. And so here's an example of the kennels in the green pod. And in the individual kennels, dogs are housed either alone or with other dogs. So this system is really useful because it's a natural system but with a lot of controlled elements. So we know who has to sustain contact with who, how long they've been in an environment, diet and other activities are controlled for, and respiratory disease can be quickly identified by shelter veterinarians and staff. So the research goals of my dissertation are to characterize differences in the community structure of microbiomes associated with infection and disease, determine how host demography drives microbial community, and identify ecological interactions that structure nasal microbiome composition. The general methods for my research is that I went to the Oregon Humane Society in Portland. I collected nasal swab samples from the dogs and then performed 16S rRNA gene sequencing. 16S rRNA sequencing is the most common method used to analyze the microbiome. Within the bacterial DNA, there's a gene that codes for this 16S rRNA that's utilized in ribosomes. And this gene in particular has been highly conserved because it's a necessity for bacterial protein synthesis. This means that all bacteria have this piece of gene. But the other special thing about it is that it has highly variable regions specific for bacterial species. That means we can use the highly variable regions to determine the taxonomic classification, so like the genus and species, for the bacteria that a piece of DNA came from. So we isolate the DNA, amplify it, make lots of copies, sequence the DNA, and then we can determine which bacteria it came from. So beginning with chapter 1, I look at canine infectious respiratory disease and microbiome dysbiosis. This was essentially our pilot study to first see if we could even quantify the nasal microbiome in dogs. And we also quickly noticed some respiratory disease in some individuals. And so canine infectious respiratory disease, or CIRD, is a common disease that affects the respiratory systems of dogs, causing sneezing, coughing, nasal discharge, but can lead to serious complications like pneumonia. And CIRD is caused by a number of pathogenic agents, including Mycoplasma cynos, pneumovirus, respiratory coronavirus, Bordetella, Streptococcus, parainfluenza virus, distemper virus, adenovirus, and a canine influenza virus. And if you own a dog, you may recognize some of these from the vaccines your dog gets at the vet. But there are 3 pathogens in particular that are important because they are not vaccinated against. And so for this study, we focused on the abundances of these 3 pathogens in relation to animal respiratory health and microbiome dysbiosis. And that's Mycoplasma cynos, pneumovirus, and this respiratory coronavirus. So to understand respiratory disease in these hosts, we try to take a more holistic approach to consider how both the pathogen community and the healthy microbial communities are structured in healthy and sick dogs, and how the pathogens and microbiome together determine the outcome of the host. So essentially looking for differences in these healthy and sick microbiomes related to disease. Our research goals for chapter 1 were to first characterize both the nasal pathogen community and the microbial community diversity, and second, determine whether there were differences in these communities of healthy dogs compared to those experiencing clinical signs of respiratory disease. To do this, we collected 104 nasal swab samples from all dogs every other week for 4 weeks at the Oregon Humane Society. We then extracted DNA and RNA from the sample and sent part of that DNA and RNA to the Center for Quantitative Life Sciences at Oregon State University for 16S RNA sequencing using their MySeq instrument. And then we sent another part to the Cornell Animal Health Diagnostic Center for canine respiratory pathogen panel testing, specifically interested in pneumovirus, mycoplasma, and coronavirus. We then combined the respiratory pathogen abundance data with the 16S microbiome data and compared the Shannon diversity, Bray-Curtis dissimilarity, and differentially associated taxa between healthy dogs and those experiencing clinical signs of respiratory disease. Based on respiratory pathogen panel data, we detected all pathogens at varying abundances. So here you can see Mycoplasma cynos, pneumovirus, and the coronavirus were the most abundant. They're also the only 3 not vaccinated against in our study population, and at varying abundance ranging from absent to high positives. We also see a lot of co-infection. In fact, 75% of our samples had more than one pathogen detected, and only 21% of samples were considered from symptomatic dogs. So this tells us that pathogens may be commonly circulating microbes, regardless of whether they cause disease. We then looked at which pathogens of the three we're interested in were associated with symptomatic dogs. So we found no difference in the abundance of coronavirus or pneumovirus in sick and healthy dogs, but we did find increased abundance of mycoplasma in sick dogs compared to healthy dogs. So this tells us that increased abundance of Mycoplasma cynos is implicated in the development of signs and symptoms of respiratory disease. We also compared the alpha diversity of samples with varying abundances of our pathogens. To determine alpha diversity, we use the Shannon index, which takes into account the number of different species and how evenly the populations are spread among the different species. We found no difference in Shannon diversity based on coronavirus abundance, but we did find decreased Shannon diversity with high pneumovirus abundance and high mycoplasma abundance. So the abundances of this virus and bacteria are related to the diversity of an individual's microbiome, and they seem to decrease an individual dog's diversity. Next, I looked at beta diversity, how the composition of taxa compared between samples and based on health status. We found a clear clustering and significant difference in Bragg-Curtis dissimilarity based on sick dogs and our healthy dogs. We also found that the dispersion of the symptomatic group was greater than the healthy group, indicating that the healthy microbiomes are more similar, but when you're sick, your microbiome changes, and that may not necessarily be in predictable ways. So our conclusions from chapter 1 are that Mycoplasma cynos is associated with disease and differences in the nasal microbiome diversity. We also saw dysbiosis of the microbiome based on health status, and pneumovirus was associated with decreased Shannon diversity. From chapter 1, we found that Mycoplasma was a common member of the nasal microbiome at low abundances, but at high abundances, it's associated with disease. And so an interesting future research goal would be to understand what causes that shift in Mycoplasma abundance that could potentially lead to disease. And all of this together suggests that invading pathogens, both viral and bacterial, disrupt the microbiota, although the directionality of this connection isn't clear. So we aren't able to tell if a dysbiotic microbiome leads to infection risk, or if infection leads to changes in host physiology, ultimately affecting the healthy microbial flora. And that brings us to chapter 2, examining whether host traits and contact shape the canine nasal microbiome, because we can see that variation in hosts exists. And based on our previous research and what we know about the microbiome, variation exists in the healthy microbiome. So there were these points outside of this really tight, healthy cluster. And we know that microbiomes are often unique and personalized. So, and because of the close connection between the microbiome and disease and potentially leaving an individual vulnerable to disease, it's crucial we know how the microbiome is structured and what forces may perturb or change the healthy microbiome. So the host itself may be a force of change regulating the microbiome through contact with other microbiota sources or demographic factors that can influence the composition of bacteria. So coming back to our meta-community framework, each individual that's viewed as its own island or patch of habitat is occupied by its microbial assemblages, and the assemblage of one individual's community may be impacted by forces like dispersal and host differences. So we hypothesize that dispersal occurs at higher rates when individuals have sustained contact with others, measured through time spent in the shelter or whether they lived with other dogs. We also believe that host differences could act as filters, allowing some microbiota to establish while others didn't, ultimately affecting the microbial community composition. These host-associated factors could be age, related to assembly. Maybe young dogs just haven't encountered many different bacteria yet, or related to immune function, sex differences, or body condition related to non-respiratory concerns. So our research questions were, what host demographic variables were associated with differences in the healthy microbiome, and does contact between dogs increase their microbiome similarity? To do this, we sampled 108 dogs as they arrived through the transfer program, immediately as they arrived, and then again every 24 hours to test for microbiome changes over time. Following similar methods to Chapter 1 for lab work and data sequencing and processing, we then tested the effects of demographic and contact variables on the diversity of samples. We first found clear compositional differences in the microbiomes of healthy dogs based on age. So young dogs are defined as those less than a year old, and adults are those over a year. First, we can see the massive amounts of Maraxa. So that's this teal color, and we can see that pretty much dominates almost all of the samples microbiomes, regardless of age. But we can see here that in a large chunk of young individuals, Haemophilus is a prominent taxa, which is not the case for adults. So that's an orange color that we don't really see that much in adults compared to young individuals. And I also want to point out that the pink, most of the pink you're seeing down here is actually mycoplasma. And we know from our previous research that mycoplasma overabundance can be associated with disease. However, all of these samples are from healthy dogs, supporting the idea that mycoplasma is a common member of the microbiome. Age was the host demographic trait most associated with differences in the healthy microbiome. So here we see some visible difference in beta diversity based on age, and some potential clustering of our young microbiome similarity versus adults. And finally, we compared whether samples were more similar based on contact. So we first classified the different types of contact. None means that any two samples never had any contact over their time at the Oregon Humane Society. Origin means that they came from the same shelter before arriving at the Oregon Humane Society and could have had some contact or microbiome sharing there. Truck contact, if they arrived on the same day in the same truck. So remember, they travel for sometimes over 8 hours in these close quarters in the truck while being moved from their California shelters to Oregon, and that could lead to some microbiome similarity. Pod means that they're housed in one of those 4 rooms or pods that I described previously. And kennel contact is whether samples came from dogs sharing a kennel. So this is the closest form of contact any two dogs can have living with another dog for several days. And then self-contact is comparing the similarity between samples collected over time from the same dog to determine whether individuals changed over their stay at OHS or whether they had an identifiably unique microbiome. And then for each sample pair, we calculated the dissimilarity using Braid-Curtis dissimilarity again. So for all sample pairs, they can be classified as having one of these types of contact. To test whether the effect of contact was different based on age, we calculated the sample pair dissimilarity for young samples and adults separately. So here we see the distribution of similarities for samples from young dogs that had no contact and adults that had no contact. So this is the range of dissimilarity for samples that had no contact, and young dogs and adult dogs. Then we compared the dissimilarities of samples with increasing forms of contact to dogs in their age group that had no contact. So we can compare for adults and young dogs whether the effect of coming from the same shelter led to their microbiomes being more similar than dogs that had no form of contact. And we found that there was no difference in microbiome similarity for either adults or young dogs based on whether they came from the same shelter before arriving at the Oregon Humane Society. So when we look at all forms of contact, we can see that there is no difference in microbiome similarity of samples from adults based on truck, pod, or kennel contact, and that's signified by these open circles here. However, when we look at the red bars from the young dogs, and we compare these to the no contact, we can see that the microbiomes of young dogs that shared the same truck, pod, and kennel were more similar to each other than they were to samples from young dogs that had no contact. And I also want to highlight that samples from the same dog across both age groups were more similar than samples without any form of contact. So this essentially tells us that the individual has their own microbiome that's unique and relatively stable over their time in the shelter. So our main results from this chapter are that young individuals are more similar with increased contact, but possible explanations could be that short-term contact actually matters to the young dynamic microbiome. Another possible explanation could be due to housing arrangements. So at the Oregon Humane Society, puppies tend to be housed with their litter mates. So it could be long-term exposure prior to this study, leading to the similarity we're seeing. So if a puppy has spent the entire 3 months of its life with the same 5 other individuals that it's genetically related to, we would probably expect those to be more similar. And that could still be a result of contact, just a little outside of our study scope. Or we think this could be because puppies don't have an established microbiome yet. They're still picking up species from the environment or other dogs, leading to random assembly based on whatever they come in contact with. Or the young immune system is still working to balance the recognition of the healthy bacteria while also trying to identify invading pathogens. But overall, the nasal microbiome of dogs is strongly shaped by host age and contact. And these results are particularly relevant as we explore ways to maintain the healthy microbiome. Beyond the host and pathogens, the healthy microbiome may also be structured via microbial interactions, which brings us to Chapter 3, Microbial Association Networks, because species interactions can be powerful community structuring forces. So similar to macroecological communities, the species composition and stability of microbiomes may be structured and maintained through ecological interactions between species. So these interactions could also serve functional purposes in microbiome stability, where any perturbations to the abundance of one taxa or the microbiome as a whole may leave the community vulnerable to change or instability. This instability could translate to variable host health outcomes for the host. And these interactions can be positive or negative. So for example, cooperation through sharing of nutrients available from the host, or cross-feeding where bacteria provide resources for other taxa and vice versa. But they could also be competition. So for resources, and the increased abundance of one taxa could lead to a decreased abundance of another. So studying interactions between species is really challenging in the microbiome because we can't actually observe interactions directly, but we can infer them using a network approach where nodes represent taxa and edges represent estimated interactions based on co-occurrence patterns. And additional network properties can inform us about the structure and function of the community. And one potentially useful part of these analyses are the identification of hub nodes and hub taxa. So hub nodes or hub taxa may have similar functional roles as keystone species in macroecological systems. They might have considerable influence on the overall structure of the microbiome network, and interactions and their removal or change in abundance might have significant consequences on the abundances of other taxa in the system. To describe hub nodes in more detail, they are potentially influential taxon whose abundance can determine the abundance of many other taxa in the network through their connections. So for example, if we removed this taxa, it would not only remove the links between these two groups of nodes, But if it was strongly positively associated with these other nodes, they could also decrease in abundance, having ripple effects on the rest of the microbiome structure. Alternatively, if we remove this node with a weak connection to the abundance of that more central node, the overall structure of the microbiome probably won't change very much. So most nodes are still connected, and the abundance of the taxa it was connected to might not change because it was such a weak connection. And one way to determine whether nodes are hubs is by using network centrality metrics. These metrics can tell us which nodes taxa have the most influential connections to other taxa in the network. So degree is simply the number of connections a node has to other nodes. Here, the node with 7 connections has the highest degree. Closeness centrality is calculated from the sum of the shortest path lengths between a node and all other nodes in the network. And therefore, it's a measure of how close that node or taxa is to other taxa, where the larger the closeness centrality, the shorter the distance between any 2 nodes. So here, node B has high closeness centrality because it has a lot of short connections to other nodes. And betweeness centrality measures the degree to which a node lies in the shortest path between 2 other nodes. So a node with high betweenness centrality would connect many other nodes and therefore be important as a hub node because its disappearance from the network could have a consequence on the shortest paths for many other taxa. So here we can see the node with the highest betweenness centrality is important to network function because if this were removed, it would disconnect these 2 larger clusters of other nodes. And eigenvector centrality is essentially the measure of the influence of a node on the network that considers direct and indirect connections, where connections to other important nodes leads to a higher eigenvector centrality. So this node here has the highest eigenvector centrality, and we can see it's connected to many other highly connected or influential nodes. And beyond network analysis, we can also use universal dynamic models to detect whether the ecological parameters of inter and intra-species interactions and growth rates are universally found across all individuals, or whether those parameters are individualistic. So the idea behind universal dynamics is that if 2 communities have the same species assemblages and the same abundance profile, then the 2 communities have the same microbial dynamics. In this way, communities become more similar only if the dynamics are universal. But there are 3 possible scenarios for community dynamics in this model. So first we have the individual dynamics. Here the assemblages and interactions would be host dependent and unique among the different communities. Or the parameters could be described as group dynamics, where microbiome dynamics can be classified into groups that your community follows, one of any number of different groups. Or the dynamics can be considered universal. So here we can see the local communities between individuals are different. These local communities are different, but the way the interactions would be described based on their abundance profiles show that species dynamics would be the same. So for example, if species A always has high abundance when found with species B across all individuals who have those species, that would support the existence of universal dynamics structuring the microbiome. Universal dynamics also state that if you plot a measure of relative abundance like dissimilarity against the species an ecosystem has in common or overlap, they predict a negative slope. So the method used to determine whether dynamics are universal are dissimilarity overlap curves. And dissimilarity is measured as the Root-Jensen-Shannon divergence to determine the similarity between 2 samples. And the overlap is measured as the relative abundance of shared species. And the calculated overlap and dissimilarity for all sample pairs represent each point in this dissimilarity overlap plane. So for example, these 2 samples here look very similar. So they have the similar species and they're found at similar abundances. And so they would have high overlap and low dissimilarity. based on the shared species and their abundances. And they would be plotted in this portion of the graph. The slope of the DOC is used to tell whether universal dynamics are supported. So a negative slope in the region of high overlap suggests the existence of universal dynamics. So this region of high overlap and whether there's a negative slope here. This is compared to the randomized data where we would expect to have a flat curve in the high overlap region. And this is because as overlap increases, we expect samples to become more similar if the microbes follow universal rates of interaction. And we can use statistical tests to determine whether there's evidence for universal dynamics as well in this region of high overlap. We can calculate the fraction of data points for which the DOC displays a negative slope and the fraction of bootstrap realizations with non-negative slopes. So our research goals for chapter 3 were to identify microbial interactions in hub taxa in the healthy canine nasal microbiome and determine whether co-occurrence networks are dependent on age and time spent in the shelter. And finally, detect whether universal dynamics of the canine nasal microbiome are supported. The methods we use for network analysis were following this paper by Stephanie Peschel et al. And the NetComi package in R. The input is the healthy microbiome count data from chapter 2. Then the network is constructed using a speak-easy algorithm that determines a conditional dependence between two taxa, so if two taxa are associated. And after constructing the network analysis is performed to obtain metrics on centrality, hub nodes, clustering, and other global network properties, followed by network visualization and comparison. Methods for universal dynamics follow methods outlined in this paper by Bashan et al. And again, using the chapter 2 healthy microbiome count data, overlap and dissimilarity are calculated for each sample pair placed in that dissimilarity overlap plane, and a curve is fitted, and then test statistics are calculated and visualization of the DOC. From network analysis, we identified Moraxilla, Bacteroides, and Prevotella as hub nodes in the entire nasal microbiome network. And while Moraxilla is the most abundant taxa in the canine nasal microbiome, Bacteroides and Prevotella were more unexpected hub nodes because they're found at relatively lower abundances. We then compared the microbiome association networks of just young individuals compared to adults and found Bacteroides again as a hub node, but just in the adult network. And based on network metrics like modularity, connectivity, and path length, we found support for the adult microbiome having more or stronger taxa connections compared to the young network, potentially meaning the young microbiome is less stable than the adult microbiome, or suggesting that microbe interactions may be less important as a force structuring the young microbiome. We also looked at how time spent in the shelter affected the microbiome interactions when comparing samples from just adults on their first day and their third day in the shelter. We found Prevotella as a hub node on day 3 and a significant difference in the sets of most central nodes based on all of the centrality measures. So this supports that time spent in shelter may impact how these microbes are interacting or the network structure as a whole is changing. We then compared microbiome association networks on day 1 and day 3, and we can see Bacteroides again as a hub node on day 1 in just the young dogs, but found no other differences in the young network between days. So this could support that the young microbiome is either very stable and we don't see any change over time, or it could be that these interactions aren't that strongly structuring the young nasal microbiome. Next, we tested whether universal dynamics appear to be structuring the microbiome across individuals. And remember that downward slope in the region of high overlap suggests universal dynamics. And so here we see that negative slope expected if universal dynamics is happening when compared to the randomized data where the curve is relatively flat in the high overlap region. We also found a very small fraction of bootstrap realizations that had non-negative slopes. So this altogether supports the existence of universal dynamics when looking across samples. But when we compare, when we separate out adult and young samples, we see that in adults, again, there's evidence for universal dynamics, as shown by this negative slope in the region of high overlap, and none of the bootstrap realizations had non-negative slopes. But in young dogs, we see a flat curve in this area of high overlap, suggesting that the dynamics structuring the young microbiome are not universal. When looking at the age groups again over time, we found universal dynamics in day 1 and day 3, although the p-value on day 3 was a little larger. That could suggest that time spent in the shelter weakens the strength of support for universal dynamics, or it could be reduced statistical power from sample size. When looking at young dogs on day 1 and day 3, we find no evidence for universal dynamics. We don't see that negative slope in that region. And so this supports that species interaction rates are not consistent across individuals, and that composition and abundance profiles for young dogs are more individualistic. So based on network analysis, we found network properties in young dogs associated with instability compared to adults, and evidence that these microbial interactions may be more important in structuring the adult microbiome. One surprising result was the identification of Bacteroides multiple times as a hub taxa, despite its relatively low abundance in the microbiome. And future research could look more into that association between Bacteroides and the abundances of other bacteria. And we also found that universal dynamics was supported in adults, but not necessarily in young dogs. And one hypothesis that could develop from this is that the young microbiome is more dynamic and changing, whereas the adult microbiome is more stable and established. So putting it all together, to summarize my dissertation results across all chapters, complex interactions exist in the nasal microbiome between pathogens, the microbes, and the host. And pathogens are associated with disease and dysbiosis. However, variation exists even within the healthy microbiome. And for this healthy variation, age seems to be a very important force. Contact also may be important, particularly for young dogs, and evidence suggests that their microbiomes may be more diverse or dynamic than an established adult microbiome. And the role of the microbial interactions on the microbiome structure looks more important and universal across adults, with less evidence for the role of interactions in young dogs. And finally, the key players in the canine nasal microbiome include mycoplasma, common in healthy individuals, but found at an overabundance in sick dogs, Moraxilla, the most abundant taxa found across all individuals, Haemophilus, found at high abundances in young dogs, and Bacteroides, identified as a potential keystone taxa multiple times through network analysis, linking it to the occurrence of other bacteria, regardless of its relatively low abundance. And thinking of the big picture, hopefully my research contributes to how we can understand microbiomes as ecological systems, and how the nasal microbiome in particular is related to health, beyond just dogs. And in the future, I think it'd be really interesting to explore the directionality of pathogen invasion and microbiome change, and also examine the long-term changes over an individual's lifetime. Overall, the importance of the factors in structuring nasal microbiome of dogs needs to be considered as we move towards the development of things like prebiotics, probiotics, and personalized microbiome-mediated treatments. So with that, I'd first like to thank my advisor, Ben. Thank you for taking me on as your first grad student 6 years ago. I appreciate all of your guidance and mentorship, and could not be more grateful for everything. I'd also like to thank my committee members, Anna Jols, Bree Beechler, Becca Terry, and Clint Epps, for providing constructive feedback at all stages of my PhD, and all of you combined making me a better scientist. And thank you to my lab group, Noah, Rachel, Isak, and Justin, for all of your help on various projects and parts of my research, and for just being great people to work with. I'd also like to thank the Oregon Humane Society, for allowing me to literally live in their animal shelter for weeks and collect samples from their dogs. I'd like to thank all of the dogs who happily and maybe sometimes unhappily participated in my research. I'd like to thank Drs. Farrell and Ottoman for their help in designing these research projects, and also thank Mia and Chris and Dr. Miller for helping me while I was at the Oregon Humane Society. And thank you to Erin Archer and Hannah Madison, undergraduates who helped me play with the puppies. Collect samples. And thank you to my family, my mom and dad, and my sister for always supporting me. And thank you to all of my friends for supporting me and making grad school in my life so amazing. That's Nicole, Haley, Caroline, Sarah, Rachel, and Anne for helping with my science so much, and Angelica and June. I appreciate all of your support and friendship over the years. And finally, I want to thank my partner, Charles, for his support, proofreading, helping with my resume, presentations, and even helping me collect samples at the Humane Society. Thanks for everything and all of the fun outside of grad school too. I don't think I could have done this without you. And with that, any questions?
So before we take questions, could we all, is it possible for everyone to unmute and give Liz a round of applause?
I don't know if they can.
Maybe they can't. Maybe I just have
webinar.
to scream on behalf of everyone. All right, well.
Yay, thanks.
All
Thanks,
right.
Ben.
And now we can take questions.
So I think Noah had one in the Q&A. Do I have any intuition about what might be happening in the younger dog's immune systems or factors only present in young dogs that would cause them not to display universal dynamics? So yeah, I think that, well, basically the microbiome develops over your age or as you age, and that's been found like across species. So I think what could be happening is like, it's maybe this taxa for the first time is like just being identified by the immune system, and it's like just making that connection, like allowing that to become a commensal or a mutualistic relationship versus like identifying it as a pathogen, and kind of getting rid of that. So it could be that just like building of that mutualism over time, and it just takes time. And I think it could also potentially be that, you know, different dogs are interacting with in different environments. And so it could just depend, it could be unique and not display universal dynamics because what that person has is not at all what I have, and like the bacteria haven't figured out how to live together with the host immune system. So something probably very complex between those things.
Other questions for Liz? I hear that the queue, okay, good.
Oh yeah, okay. So at what depth were the swabs inserted? So I think it was like about 2 centimeters. So it was just the like very anterior part of the nasal canal. Yeah, so it was like fully inserted and then like moved around for at least 3 seconds to be considered like a good swab sample, which sometimes the dogs would not allow to have happen to them.
Other questions?
Anything else?
Ricardo says, oh, here we go.
Oh yeah, okay. So mycoplasma was more common in, oh no, it disappeared.
Ricardo,
Okay.
can you put the question back?
I found it again.
Okay.
Okay, so mycoplasma was more common in less healthy dogs. Do you think it's related to those dogs being vulnerable due to their microbiome, or their microbiome changing as a result of physiological stress? So it's tough, and I don't really know. I think that I would bet it was more that something, yeah, like the stress of the environment could make the dog like more vulnerable to the already established mycoplasma kind of just taking over. And then that, like, I remember that mycoplasma can have an effect on like mucociliary clearance. And so if there's an overabundance, that might start having a negative effect on their health. But it is really hard to say. And that was kind of an inspiration for the second chapter, or looking over time, but unfortunately are also good for the dogs. They get adopted really quickly. So I couldn't really tell if there was like any change that made them more vulnerable based on like their original microbiome. But that's a really good question. And that's like a question the microbiome is still so fascinating, because I don't know if it makes you more vulnerable to disease or something just happens and mycoplasma takes over. Do I think the host is regulating the composition of the microbiome, the taxa present in regards of ecological interactions in order to obtain such universal interactions? Oh yeah, okay, so they could be. The host could be regulating through like immune system function. If something starts getting too abundant, it's like, no, I don't want that to have to happen, to get that kind of universal interaction. And I think maybe that could be a possible explanation for why in young dogs that's not seen. So if their like immune system hasn't developed fully yet and led to that mutualism. And then would I expect similar results if I looked at the gut microbiome of dogs or not? Yeah, I think I would expect. So a lot of research has been done on the gut microbiome relative to the nasal microbiome. And I think there's some things unique about the nasal microbiome, like it's has a lot of contact with like the external environment and maybe more variable because of that. But I think and hope that the patterns found in the nasal microbiome would be similarly found in the gut. So I think so, yeah.
Can I ask a question?
Sure.
Hey, Liz, where'd you get the awesome microbe art?
Oh, thanks for asking. I made it. So I drew this like this background here and those symbols that you saw. And then some of my friends' dogs donated their pictures to some of that work. And a lot of the pictures of the dogs were from the dogs in the study. Yeah. So I also love scientific illustrations and drawing cool science art.
Thanks. Any other questions? Yeah, thanks everybody. Any other questions from the audience? Soon we'll move to the closed door session with just Liz and the committee. Are there any other questions before that? If not, let's thank Liz again. And then before we do that, committee, you should have received another Zoom link for the closed door session, and just email me if you are having trouble getting in. All right, congrats, Liz. Thanks for the talk. Woo!
Yeah, yay. Thanks everybody for coming and your questions.